Okay, fast forward. 3 individuals. These are 3 individuals that should have won a Nobel Prize but never did. They were completely passed over for the Nobel Prize, but they carried out one of the greatest experiments in biology of all time. These individuals, Oswald Avery, Colin MacLeod, and Maslin McCarty, discovered that DNA was the genetic material. And if that doesn't deserve a Nobel Prize, I don't know what does. That was one of the most important experiments, and that was one of the experiments that swayed Watson and Crick that genes were made of DNA, because they knew of the experiments of Avery, MacLeod, and McCarty, which were carried out in the mid-40s. And when you read the double helix, you're going to read a little bit about those experiments. Who was Avery? Who were MacLeod? Who were McCarty? They were all physicians. All of them were working at the Rockefeller Institute, now the Rockefeller University in New York. All of them were interested in flu because, again, it's still a killer and still there's no cure. And so what they were trying to work on was, one, what was the determinants of virulence in the Streptococcus bacteria? That's what Avery's sort of known for. He discovered that the sugar coat of the bacteria is made out of sugar. And he discovered what sugars actually are responsible for the virulence. That was one of the things that he did because he was a remarkable biochemist and microbiologist. But the point here is that going back historically, they were trying to understand what the transforming principle was. Now, I have to say, and it's very difficult to be able to transmit this, particularly the people that don't spend most of their time thinking and breathing science. Oswald Avery and his collaborators had a question. What was the transforming principle? But in order to answer that question, they had to invent every single solitary technique before they could do the experiment to answer the question. Because at that particular time, the field of biochemistry was relatively primitive. The ability to isolate DNA was pretty primitive. The ability to isolate RNA was primitive. The ability to isolate molecules in cells were very primitive. And in many respects, Avery and his colleagues devised procedures, some of which are still used today, to purify things from cells so that they could go after and answer what was the transforming principle. So it wasn't like they just took techniques off the shelf or read them in a journal and used them in their lab. Because they had a question, and this is true genius and it applies today, if you have a question and there's no answer and no technique, you invent the technique. That's the bottom line. And that's where real creativity in science comes about. And so what Avery et al. Did was ask a number of questions. They asked the following questions. Remember that Griffith's experiment was carried out with a mouse. So the transforming principle could have been a property of the mouse. Didn't necessarily have to be a property of the bacteria because this mixture of heat-killed smooth and rough was injected into a mouse, and the mouse could have contributed something, which was the transforming principle. So they wanted to eliminate that possibility or determine whether it was a possibility. And once they determined that by experiments that I'll show you in a second, once they determined that it was the bacteria cell itself that was the container of the transforming principle, then they wanted to know what that molecule was. And then by the process of elimination, as you will see, they were able to determine that DNA was the genetic material. And so what were those experiments and how were they done? So here's the first simple experiment. The first thing is they wanted to demonstrate whether transformation could occur. You with me, Dora?
Yeah.
Am I confusing you?
No.
Good. They wanted to determine whether transformation could occur in the absence of the mouse. So they just used test tubes and culture plates, sort of like what I just showed you. And so what Avery et al. Did was take the living form of the rough, the avirulent form, which, remember, you can distinguish them by their morphologies, took the heat-killed virulent form, they mixed them together in a test tube, no mouse, waited a while, plated them out on a plate, and lo and behold, they got living smooth form. So that showed that the transforming principle was a principle that was part of the bacterial cell, and you didn't need the mouse. It was a very important experiment. But the next thing is that they wanted to know what molecule inside the bacterial cell was the transforming principle. And keep in mind, it could have been anything. Could have been anything, because here are the components of a typical bacterial cell. Mostly water, ions, sugars, amino acids, et cetera, and significant number of macromolecules. Now, what are these things called macromolecules? Well, they're very large molecules, huge, like DNA. And these macromolecules you know about. They're carbohydrates, they're fats, they're proteins, they're nucleic acids. They all have a similar kind of structure. They consist of units, monomers that are bonded together into big, long, repeating chains of that monomer. So a protein has amino acids that are bound together. A nucleic acid had nucleotides that are bound together. Lipids have fatty acids. And polysaccharides and carbohydrates have sugars. So Avery et al. Didn't know anything except there's a lot of stuff in these cells. And any of this stuff, it could have been a sugar, it could have been fatty acid, it could have been a protein, it could have been a nucleic acid, be the transforming principle. So here's the experiment that they did. Now I want you to think about this carefully and critically, and I don't want you to look ahead on your handout. So what Avery et al. Did was learn how to purify from these virulent bacteria all of these macromolecules. And as I said, they invented the techniques in order to do that as they went. And then what they did was they took purified macromolecules, RNA, DNA, carbohydrates, fats, and they mixed them one by one in a test tube with this avirulent rough strain. And then they looked to see what part of the biochemistry of the cell could transform rough into smooth. And lo and behold, they found that when they added RNA, nothing happened. When they added DNA, they got transformation. And mind you, theoretically, these were pure molecules. So the result was that when they had a purified DNA fraction and added it to the live rough form of this bacteria, the cells were converted to virulent smooth. How many would say conclusively that that experiment shows that DNA is in fact, I turned it off. Let's just take a vote. How many would say conclusively that that experiment shows that DNA is the genetic material? Yes or no? Eden, yes? How many say no? How many don't know? Thomas, you didn't vote. Let's vote. How many say that when we take the DNA from the smooth form, mix it with the rough, rough is transformed into smooth, that DNA is the genetic material? How many say that shows that DNA is the genetic material? Yes or no? Raise your hands high. How many say no? Okay, Rose. Why do you say no? Ingrid, could I speak to you a minute? Go ahead, I'm listening.
Well,
Where's the air conditioning in this?
I don't actually know conclusively, but I'm going to say no just because
Yeah, go ahead.
I feel like, well, not feel, but
Tell me why. Look at me and don't look down.
just because
What may be a problem with that experiment, Rose?
that it could be some other thing in the, no, that doesn't make any sense.
Well, it makes a lot of sense. What do you think, Dora? Do you agree that that shows that DNA is the genetic material?
I thought it did because he tried. Well, I guess there are other tests he can run with
Okay.
different. Oh, okay, but then when he
Remember,
tried
this
with
is
the
1941. So
Oh, then
what
I
could
would
be a
think it
problem
was-
with that experiment? I want you guys to think about scientists here. Remember, scientists don't believe things. In fact, they try and find reasons for why something is not correct. That's how we think as scientists. We look at a result and we say, what's wrong with that result? And we try and come up with alternative explanations for the result. We rarely accept the result on face value, like all of you did in the room. DNA is the genetic material. Let's go write a publication. What do you think? Well, Eden, you've spoken already. I want to hear from somebody else. Albert!
Well, they don't know, like, for sure.
What's your hypothesis?
Well, that no.
Speak in.
My hypothesis that possibly there could be.
What's an alternative explanation?
Well, it could have not been completely purified yet.
Say that again.
It could have been not completely purified.
Yeah. Would you have been able to detect 1 or 2 protein molecules in that prep
No.
in 1941?
No.
I don't think so. They didn't have mass spec and all the sophisticated. So it could have been, there could have been a contaminant.
Yep.
Okay. Well, that's exactly the way they thought. They got this result and they said, what could be other explanations? And instead of saying that DNA was the genetic material from that experiment, they said, we better do another experiment to try and show beyond a shadow of a doubt that DNA was in fact the genetic material. And so they did the following experiment. There are these things called enzymes, which you know about, like restriction enzymes. But enzymes are very specific. An enzyme that degrades DNA only degrades DNA. An enzyme that degrades RNA only degrades RNA. An enzyme that degrades protein only degrades protein, so on and so forth. So what would you predict, Anna, if you may have thought that DNA was the genetic material, but Albert said it was contaminated with protein. Therefore, maybe protein was the transforming principle. What would you say if you treated that DNA with this enzyme that destroyed it? If it was DNA that was the genetic material, would you get transformation or not? Let's say that you think that DNA is the genetic material, but Albert said, as people did at that time, that it's contaminated with, let's say, protein.
Okay.
And let's say you said, okay, I'm going to prove to you it's DNA because I'm going to take this enzyme that degrades DNA, and when I treat the DNA with this enzyme and then add it to the rough cells, what would you predict would happen if DNA was the transforming principle? Would you get transformation or not?
And you treated it with the enzyme?
That destroyed the DNA.
Then you
What
would.
do you think? Your experiment.
I think that if it was DNA, that I wouldn't get the.
Yeah, so if you thought it was DNA and you destroyed the DNA, you wouldn't get any transformation. But if Albert was correct in that it was a contaminant with something else, if you destroyed it, you'd still get transformation. Mm-hmm. And so Avery et al. Did the following experiment. They learned how to extract these enzymes. They learned how they worked. They took each of the molecules of the cell, protein, DNA, RNA, fats, etc. They mixed them first with enzymes that would destroy them, so to speak. And then they looked to see what would happen in the transformation. And the only thing that produced essentially a negative result was the fact that when they destroyed DNA, they got no transformation. Meaning when they destroyed protein, they still got transformation. Why? Because the DNA was still there. When they destroyed RNA, they still got transformation of rough to smooth because DNA was still there. When they destroyed the fats, et cetera, et cetera, et cetera. So the point here is this is the critical experiment. Never accept a result at face value. Always have an alternative explanation in critical thinking. And so, as Albert said, the initial experiment suggested that DNA might be the genetic material. But on the other hand, there might have been an alternative explanation that it was contaminated. And therefore, when they took an enzyme, which was specific for DNA, they destroyed the transforming principle. Aichi, would you take this back to the lab, put some new batteries in it for me? Thank you. And so we have a remarkable experiment. And Avery, McLeod, and McCarty published that experiment in this journal of, I believe it was British Medical Journal. They published it in 1944, and they determined and said that DNA was the genetic material. Kathy Zhao, Do you think that Avery et al. Said that DNA was the genetic material from this experiment in their publication? Would you have said that? What do you think?
I think yes.
I can't hear.
Yes.
Well, in fact, they were very cautious. And they said, if these results are repeated, then DNA has a biological activity of which we are yet to understand. So they didn't come out and say DNA was the genetic material. They sort of beat around the bush because they were cautious, because this was a monumental change in thinking of the time. But it turns out it stood the test of time. And so what that said was that naked DNA of the smooth gene was spewed into the environment by the boiling and killing of these cells. And Chris, you can boil the DNA and it won't destroy it. You'll still have some stuff left. It's very, very, very responsive to high temperature and refractive against being destroyed. And then that DNA was taken up by the live cells, incorporated into the chromosome of the live rough cells, and then specified a particular trait, meaning a smooth trait, which in fact was the virulence trait. And so we have sDNA taken up by the cells, the S gene transcribed into smooth mRNA, the smooth mRNA translated into smooth sugar coat protein, and that protein helped construct this capsule, which protected the bacteria from the antibodies. They looked smooth, and they were protected. Tiger. Where's the microphone? Go ahead.
Can raw DNA be taken up into cells very easily then? Because every time they mixed it in the test tube, all the cells were transformed?
In bacterial cells, yes. In bacterial cells, yes. And in eukaryotic cells, yes, because I showed you a whole bunch of experiments last week. I mean, that's able to do that, but we had to give it some assistance in order to do it. Bacterial cells take up DNA from the environment all the time. And in fact, it's one of the processes by which some bacterial cells generate genetic variability in populations by exchanging naked DNA, which is in fact in the sewers. Not all bacteria, but this kind of bacteria has the ability to do that. Samantha.
Okay, so my question is, you heated up the smooth DNA to see if once you mixed it with the rough, but if you killed the smooth bacteria, but does the DNA
But there's no bacteria in these experiments. They're all gone, remember?
back with the mice?
All gone, yeah.
I'm just wondering, when you heat up DNA, does it die or does it stay alive? Can DNA
How
die?
can DNA be alive?
So DNA can't.
What is your definition of alive?
Has living cells.
What's your definition of being alive? What are the properties of life?
Respiration?
Does DNA respire?
No.
Are you going to, let's not have a memorized list of A, B, C, D, and E. I don't think DNA is a molecule. DNA is not a living cell. So DNA can function, but it's not alive or dead.
Okay.
Remember what Watson said in the movie, in Crick, that DNA is immortal, which in fact it is.
Okay.
All of us, in fact, have some DNA that went way back with our ancestors. It's always there because it replicates. So there's no live or dead. You can't think about it that way. Any questions, Dora, Faria? Is it clear? Any questions? Okay.
No.
Go ahead. No or yes?
No.
No? Okay. So how does this then satisfy the predictions of a molecule which is the genetic material? Well, first, the smooth DNA was able to replicate because it got in the rough cells, and it was perpetuating that phenotype from generation to generation because the smooth cells now could produce more smooth cells, so the DNA had to be replicated. Secondly, does it be able to direct the phenotype? A naked piece of DNA, yes, the naked smooth DNA is able to transform the rough into smooth, so therefore it has to tell the rough cells to make now a smooth coat. So it's making a phenotype. And is it stable? Yeah, absolutely, because once you transform rough into smooth, they stay smooth for generation after generation after generation. So the predictions of a molecule of being the genetic material are satisfied by this experiment and the experiments of the glowfish and the experiments of all the things that I talked about last week. Okay. What do you think, Ben? Do you think that transformation is only limited to smooth and rough, or do you think any gene for which you have some unique phenotype can be transformed into some cell and be able to demonstrate a new phenotype? Do you think that you could take any piece of DNA and put it in a cell and be able to generate a new phenotype?
Yes.
Yeah, absolutely. So transformation is not limited to smooth and rough. Transformation can be done with any particular gene for which you have a known phenotype. And last week you discussed a marvelous transformation experiment, because now we use the word transformation, which is what I told you originated from Griffith. We use that for transformation of any phenotype in any cell with a foreign gene. So we use transformation for genetic engineering. And so here we have the Cohn and Boyer experiment. Here we have a combination of, two different antibiotic genes. You know how to do this because it was on your quiz. You combine them together, and now you get a new phenotype. The cell is now resistant to both antibiotics instead of one, and that's all done with naked DNA, all being manipulated in the test tube. And so we can, in fact, do transformation with antibiotic genes, as you know. And that was the invention of real genetic engineering by Cohn and Boyer. Now, let me digress a minute. I love talking about this stuff. It's so much fun. Antibiotics are not antibodies. Remember that. You're going to talk a lot about antibodies tomorrow in discussion. They're not antibiotics. Antibiotics are small molecules that can be produced by microbes, bacteria, and fungi that are secreted into the environment and destroy the target organism. And they destroy the target organism either by preventing the ability of the, say in the case of bacteria, to divide. And so when you take penicillin, how many people have ever taken ampicillin or penicillin? Well, the reason why you destroy that bacteria and get better is because that antibiotic is essentially preventing the bacterial cell from making a cell wall. Without a cell wall, they just pop. They pop like a balloon. And so that's one of the targets of the antibiotic. Another target of the antibiotic is the replication of DNA. So there's antibiotics that prevent the bacterial DNA from replicating, and if you prevent the DNA from replicating, the cell is going to die. Another target of a different antibiotic would be the production of proteins. And if you inhibit protein synthesis, the bacterial cell will die. And then other antibiotics inhibit critical enzymes, let's say in the metabolism of a bacterial cell, let's say the ability for it to respire or make oxygen or just go about its normal living processes, the bacteria will die. And so the targets, as you can see, of penicillin is cell wall biosynthesis. Cell wall synthesis, penicillin. The target of tetracycline, how many of you have taken tetracycline? It's a pretty old antibiotic, but maybe some of you. Target is protein synthesis. How many of you have taken erythromycin? And the target of that is, again, protein synthesis. And then how does the bacteria combat this? Remember, antibiotic-resistant genes on plasmids. If the bacteria doesn't have the ability to combat the antibiotic, it's going to die. And so the bacteria starts to acquire mechanisms to detoxify the antibiotic which is coming into the cell, again, from the environment. The antibiotic is not produced by this bacteria. It's produced by bacteria that want to kill it or fungi that want to kill it. Bacteria wants to kill the fungi protects itself with an antibiotic against the bacteria. A bacteria wants to kill another bacteria, that bacteria protects itself against the antibiotic. It's all warfare, constant microbial warfare going on. Offense, defense, offense, defense. Jessica, microphone.
Okay, if all of them,
I don't know what them is, and I don't know what off is. Can you be more specific?
if all of the antibiotics serve the purpose of destroying the bacteria, how do they determine which one is used for what disease? I mean, for what a treatment.
Yeah. So the question is, if all of the antibiotics are involved in destroying bacteria, how do they determine which one is useful in treating a particular disease? And there's a very simple answer to that. It's what the bacterial disease is not resistant to. In other words, if you had essentially the beginning of antibiotics, there were very few diseases of bacterial origin which had bacterial resistance. So bacterial resistance has been built up in human populations because of the use of antibiotics. And so because plasmids move from cell to cell, these plasmids can carry an antibiotic resistance gene for penicillin into a population. Now all of a sudden you might have a bacterial disease like pneumonia, and it's no longer able to be treated with, let's say, penicillin. So now you use tetracycline because they have different targets in the cell. So it's just trial by error essentially. And these antibiotics are specific for bacterial cells because the details of these processes that they intersect are specific for bacteria. Now, how do we get resistance? Well, here's where the plasmids come in. So the plasmids have antibiotic resistance genes that evolve to keep the bacteria alive. And how do they work? Well, one might be the evolution of a bacterial gene which makes a little pump that pumps the bacteria, which pumps the antibiotic out of the cell. So the antibiotics get in, and then they get pumped out as soon as they get in, so the bacterial cell is now still resistant and is not killed by the antibiotic. Another way is that we have a protein that is encoded by this plasmid antibiotic resistance gene, and that particular protein binds to the antibiotic and alters it inactive by some chemical reaction. And then we have another process here where we essentially have a bacterial enzyme. Let's say that enzyme is the target of the antibiotic. The bacteria changes the structure of the enzyme slightly, its own enzyme, so the antibiotic can't affect it at all. So those are three different kinds of mechanisms which can result in antibiotic resistance. Samantha.
Are these all like mutations that
All
adapted
mutations.
that adapted into?
Yeah, they're all spontaneous mutations, because remember, any gene in any genome can mutate spontaneously. But if you have in a population a spontaneous mutation, which happens to be, let's say, altering an enzyme spontaneously, that's the target of an antibiotic, that cell now has an advantage because the antibiotic won't kill that cell. It kills all the other cells. You have Darwinian evolution right in front of you, and now there's a selective advantage of that cell as compared to the other ones. And now that whole population has that antibiotic resistance gene because all the other ones are killed off.
Should we not be using antibiotics anymore?
Not as much as we use them. Shouldn't have this crap that's in your drugstore, excuse the expression, that says we have antibiotics in soap and all this other kind of stuff. That's really a big no-no because it can lead to resistance. One more question on resistance and I want to move on. Go ahead.
Is that why they, when you're taking antibiotics, they recommend that you take it for the given time?
I can't hear you. Speak up.
When you're taking an antibiotic, they normally give you a time frame for which you should take
Yeah,
it.
so you want to take it in a massive dose over a particular period of time, get rid of that bacteria. If you take it as a prophylactic over a long period of time, your chances of getting resistance to that antibiotic has increased.
Right.
So you want to just kill the bacterial population and get rid of it. Okay, let's move on. That was a digression. So we can transform cells, as you know, with antibiotic resistance. We can transform cells in eukaryotes with DNA. We have the mighty mouse. And again, it shows that DNA specifies a particular trait. We can transform the mouse. We can transform the mouse with another gene. We can transform the fly and make eyes on the leg. And we can transform human beings and create new phenotypes. There's no limits. None. Zero. Zippo. We can do anything. And that's what's so exciting and daunting with this technology. And of course, we can do it with plants. And we'll talk about that next week in discussion and we'll have a guest speaker that'll talk about it next week. But again, here's all the different things that have been done by transformation in plants. We can make decaffeinated coffee by genetic engineering. We can make allergen-free grains by genetic engineering. We can make herbicide-resistant plants, viral-resistant, insect-resistant, cold-resistant, all the things that you'd want to do to improve plants for agricultural purposes. And we can make, in fact, a glow plant. So I'm not going to go over the mechanisms here of transformation. You can read them yourselves, but in fact, this is the basis of all genetic engineering right here. You have an engineered gene. It can get in a cell. It can replicate. It can specify a particular phenotype. All it does is obey the rules of what genes are. Remember I said last week, and I'm going to say it again. Whether you engineer a peanut to be a better peanut using breeding, whether you engineer the Chihuahua into a Great Dane using regular breeding, you're manipulating a gene. In the case of the classical breeding, you're selecting for one form of the gene versus the other, but you are selecting for a slightly different protein, which will function in one case compared to another. When you do genetic engineering, you can take any gene, and you know that that gene specifies a particular trait. You can put it in any organism. That gene is then taken up by the chromosome of that organism. It's replicated because it's passed on from generation to generation, and it's expressed into a particular phenotype. No hocus pocus. Anyone who comes to you, let's say your parents or your uncles, aunts, your brother, sisters, say, isn't this genetic engineering scary stuff? Goes on in your cells all the time. It's no different. Like I said, it's a natural process. Whether you do it by breeding, which you have to stay within species boundaries, or whether you do it the modern way, which is highly more accurate, much safer, much more precise because we know what we're doing, it's a normal process. It's no hocus pocus. And I'm going to emphasize that over and over and over again. Now that you know what genes do, let me tell you what they are. And this is where it's important to focus in a little bit, because I'm going to go into a tiny bit of chemistry, but not very much, because I want you to really understand how these genes work from a conceptual point of view. If you look on the screen, what you'll see is a globin gene. That's the gene that specifies the protein in your blood cells that makes your blood cells red and binds oxygen and keeps us alive. Notice that that globin gene has a unique sequence of nucleotides, A, G, C's, and T's. And notice that that gene, this is very important, so circle it on your notes, has a beginning and it has an end. And those of you who are comm studies majors and history majors and everyone else who will never hear anything about this ever again as long as you live, have to recognize that if something has a beginning and something has an end, it has polarity. I have a mouth and I have a butt. A mouth and a butt. I have polarity. I have a head and I have feet. Well, this globin gene has a beginning and an end, and the sequence goes from the first nucleotide to the last nucleotide, and it's called 5' to 3'. That is the order of the nucleotides, and you need to understand that to understand genes. In any case, a small digression. We have a 5' and we have a 3' end, and that globin gene is unique because of the unique order of those nucleotides. Unique order of those nucleotides. And therefore, the nucleotide order specifies what the protein does. Max, if I have a different order of nucleotides, do I have a different gene? No, you just have a gene. Different order of nucleotides. Where's the microphone? Do I have a different gene? Completely different order. Not just one nucleotide change, a completely different order. What do you think? Yes. Yes or no? I got it. Should be on.
Yes.
Yeah, if I have a different order of nucleotides, I have a different gene. Now, how do I have an allele tiger? What would be a different form of the globin gene?
It would just be.
Just tell me where to point.
You can point anywhere.
Why don't you point? Tell me where it would be. Yeah, you can change one nucleotide and get a different form of the globin gene, and that's in fact correct. So if we change the entire sequence, we change the gene because it makes a different protein. If we change one nucleotide, 2 or 3, we get a different form of that gene, which is an allele. Totally different sequence, different gene, slightly different sequence, 1 or 2 differences, different form of the same gene, different protein. Therefore, we have an allele. Let me continue on this line. We have a 5' end of the gene and we have a 3' end of the gene. And now I'm going to introduce some new terms. This order of the gene is relative, and you're going to understand this. I'm not going to confuse you because I'm going to go over it again, is relative to what's called the coding or the sense strand of the gene. Remember that the gene has 2 strands. We call them Watson and Crick. Only one strand tells the cell what to do. So the 2 strands of the globin gene in the chromosomes, only one strand tells the cell what protein to make. And that strand which tells what protein to make is called the sense strand. And so the sequence of the gene, 5' to 3', is relative to what we call the sense strand, or the strand that specifies the protein. And that's going to make perfect sense to you in a few minutes. Okay, so we have a gene. We have the beginning, the 5' end. We have the end, which is the 3' end. And we have order of nucleotides in the middle. And that's what specifies the unique gene. More than that, that's what specifies, Marvin, a unique genome. Do you think that the sequence of your genome differs from the sequence of a plant genome? Yeah, absolutely, because that's what confers biological uniqueness, the sequence of these nucleotides. Might be the same nucleotides, but they're in different order. And that different order specifies different genes, different proteins, different functions, different traits. The gene that specifies red and yellow tomatoes is different from the gene that specifies pear-shaped tomatoes from big round tomatoes, because they have a totally different DNA sequence. But the gene that specifies yellow and orange tomatoes, that gene has different forms, the yellow form and the orange form, and those forms, or red form, and those are just different forms of the same gene, or alleles. And it's important to keep this straight because we're going to be talking about this in the context of DNA fingerprinting and forensics and all of that different stuff. So the point here is if I know this DNA sequence and I can synthesize it, which I can do anything I want. And that's in fact true using genetic engineering. And you saw the ultimate result of that, of putting an eye on the leg of the fly in addition to the head. Okay, now I'm just going to go over this briefly, and I'm not going to spend a lot of time on this because it's relatively straightforward, and I'm going to conceptualize it. Genes are made up of DNA, as you know, and DNA is made up of nucleotides. And this is about the only chemistry you're ever going to get in this class, but I want you to understand it. But I think all of you have had this at some point in biology, but it's pretty simple. But I'm going to give you Bob Goldberg's version, which you're not going to hear anyplace else. The building block of the DNA molecule is the nucleotide. And the nucleotide consists of a sugar, a base, and a phosphate. Sugar, base, and phosphate. That's what a nucleotide is. So an adenine has a sugar base and phosphate. Thymine has a sugar base and phosphate. Cytosine has a sugar base and phosphate. Adenine has a sugar base and phosphate. All these nucleotides have these particular structures on them. From the point of view of a gene, here's what's important conceptually. You with me, Nancy? You with me, Anna? Remember I said 5' to 3'. That's the order of the nucleotides. The sugar, here's the sugar, and I'm going to show you this more in detail in a second. This sugar has polarity. Notice that in one part of the sugar is a phosphate, another part of the sugar has, let's say, nothing for now, and another part of the sugar has a base. Everything joins together in the nucleotide via the sugar. The sugar has 5 carbons. 5 carbons, each numbered 1 prime, 2 prime, 3 prime, 4 prime. Those are the carbons of the sugar. At the 5 prime carbon binds the phosphate. At the 3 prime carbon of the sugar is just a molecule called a hydroxyl OH. Just the only chemistry you're ever going to have, but it's important to understand. The sugar has polarity. The carbons of the sugar show polarity. At the 5 prime carbon of this sugar in the nucleotide, the deoxyribose sugar, we have a phosphate. We specify polarity immediately. At the number 3 carbon of the sugar, we have this OH, indicated by nothing here. And essentially, at what is called the 1 prime sugar carbon, we have the base. The point here is, I don't care if you remember 1 prime, 2 prime, 3 prime, 4 prime. I don't care about any of this. What I care about is simply that the sugar is the hub in which the base, the phosphate, and the sugar comes together to make the nucleotide, but there is polarity. And the polarity is specified by phosphate at the 5 prime carbon, hydroxyl at the 3 prime carbon. We now have polarity. The beginning of every single solitary gene, so to speak, conceptually, is 5 prime because the sugar at the end of the gene, theoretically now, if it was the beginning of a chromosome, if it was the end of a chromosome, the first nucleotide of a chromosome going from here all the way to the end, it might have a phosphate exposed. And that dictates its polarity. If we go all the way to the end of the chromosome, theoretically, to the 3 prime end, or the 3 prime end of the gene, it's 3 prime because it has no phosphate, it has a hydroxyl. And that's how we define. Let me go on, Max, because I'm going to go over this. Let me go over this, because I'm coming back to it. I'm going to repeat it. Okay? So the point here is that the gene has a unique order of nucleotides. 5 prime, 3 prime. That's what makes it unique. It's just simply the order of these nucleotides that can then specify the order of nucleotides in an RNA, which then can specify the order of amino acids in a protein. If the gene has a specific order of nucleotides, the protein has a specific order of amino acids. Therefore, the globin gene gives rise to globin protein. The glowfish gene gives rise to glowfish protein, and so on and so forth. And so what we then have is these nucleotides joining together in a unique order on a DNA strand, 5 prime, 3 prime, 5 prime, 3 prime. Nucleotide equals sugar, base plus phosphate. Nucleotides are linked together by particular bonds, and we have a double helical structure, and the sequence of those are totally different. Let me review for you, Max. In the DNA molecule, we have purines and pyrimidines, different chemical structure. We have 4 different nucleotides: sugar, phosphate, base. Notice on the sugar, 1, 2, 3, 4, 5 carbon, phosphate at the 5, base at the 1, and this OH at the 3. That gives polarity. And notice that in the cell can recognize the bases, and it's remarkable. I know, Samantha, you might find this hard to believe, but you're really just a bag of chemical reactions. I mean, everything that we do is a specific chemical reaction because it's chemistry. And notice that we have these bases which confer the specificity of the nucleotides. Notice that there's a particular molecule attached to this purine. Notice that there's a different atom at this position, an oxygen, instead of this NH3 group at the same position. So now of this purine, we have another different group. So we have guanine versus adenine. It's just chemistry. That's all the cell can recognize. We think because we have chemistry. I move my hand because I have chemistry. I speak because I have chemistry. Everything we do is because of chemistry, including the functioning of genes. So let me carry this forward. So we have, Max, as I said, we have the sugar as the hub. We have the 5 prime, which has the phosphate. We have the hydroxyl at the 3 prime. And note the polarity. That's where the polarity of the gene comes from. It comes originally from the polarity of the sugar. Now, what's your question, Max? I need a microphone because the kids in Davis can't hear you. I want a conceptual question. I don't want a detailed question. Give me a conceptual one.
Okay. So 5' is phosphate.
Speak up.
5'
The
entails.
5 carbon on the sugar binds the phosphate.
Okay.
I want a conceptual question. I don't want to go into any details.
I just on the slide.
If you have a detail, come to me after class.
Okay. All right.
Okay? I want conceptual only because we only want broad strokes in here. Okay? Because you're not going to be asked any of this stuff. Okay? All right, now, there again is the actual structure of the sugar molecule. Notice again the different carbons. Notice that we have the phosphate attached to here, and we have the OH attached there. We have polarity. Now, how do the nucleotides get together? The first thing that's important to keep in mind is that my skin cells perpetuate new skin cells. My eye cells perpetuate new eye cells. That means the genes replicate themselves from generation to generation almost identically. That means there has to be, Ingrid, is there a problem? Do you have a question? That means that there has to be, in fact, a template. And the template means the order of the nucleotides in the gene then specifies the order of the newly replicated molecules, the next cell generation. And so what that means here is that when the DNA is put together, there's a very specific order. And the order of those nucleotides is specified very nicely. The order of those nucleotides is specified very nicely by whatever gene is dictating that process. And we'll talk more about that next week. But notice that we have in shorthand a C, an A, a G, phosphate to hydroxyl, 5' to 3', C, A, G, 5' to 3'. We have a very specific order, and that order is put together during the process of DNA replication. Okay, so we have nucleotides, we have a nucleic acid, but those nucleotides have to come together in a nucleic acid into the double helix. And remember Chargaff's rules, that arrogant Chargaff who was arrogant to the day he died. And in fact, Chargaff, of all people, when genetic engineering was invented, was one of the biggest opponents of genetic engineering on the face of this earth. He picketed and campaigned vigorously against using any genetic engineering because he thought it was against the laws of nature. Maybe that's why he didn't really understand the structure of DNA from what he did. But Chargaff's rule said purines bind with pyrimidines, and that, as you know, helped Watson and Crick solve the double helical structure of the DNA. And again, I'm not going to go over this here because you can read about it and you know about it, but there are certain things that are happening here, A's with T's and G's with C's that are complementary, and that's the basis of the replication of the gene. In other words, that each base can dictate what the sequence will be on the complementary base by DNA replication. And so we have a double helix, complementary strands, A's, T's, G's, C's. The sequence of the strands differ, bases to the interior, phosphate to the excess. DNA strands go in the opposite direction, as we discussed earlier, and the sequence of each chain automatically specifies the sequence of the other chain. And so we have the double helix, and there it is. And I'm not going to go over that. Let me wrap this up, because we have a couple things that we have to do. The first thing is that genes reside on chromosomes. So they're not in isolation of each other. They behave in many respects independently of each other, but they reside on a chromosome in a linear order. And so what you're looking at here is a human chromosome. And that's, for example, this chromosome is one long piece of double-stranded DNA wound in a little coil. We'll talk more about this at another point in class. The DNA in your chromosomes is linear. The DNA in a bacteria is circular. And what you're seeing here is the sequence of the E. coli genome, the bacteria that lives in your gut, And what you can see is two strands. They go in the opposite direction. And there are genes on both strands. We'll talk about that again next week. And furthermore, what you can see here is that these genes work independently of each other. You have gene A, which makes protein A. We have gene B that makes protein B. We have gene C that makes protein C. And why? Each gene has a beginning. Each gene has an end. And those are defined by specific sequences. Beginning, end. This gene has a different beginning, a different end. Different beginning, different end. They can function independently. They have different sequences. They specify different proteins. Each of these proteins specifies a particular trait. And we'll talk a little bit more next week of why these arrows go in one direction and go in the other. And furthermore, you can look. Here's the human chromosomes, and you can see these little green things, and those are specific genes. You can map them, and for medical genetics purposes, this is a very important technique of being able to isolate and determine where a particular gene is on a chromosome. Here's a whole bunch of genes on one of your chromosome. Here's a whole bunch of genes on the E. coli chromosome. And finally, here's what it all looks like from the sequence of the human genome. Here's one of your chromosomes, chromosome 22. Remember, chromosome 21 causes Down syndrome. Remember, chromosome 20, Down syndrome is due to an extra chromosome 21. Most embryos that are formed in humans that have extra chromosomes are lethal. It's very rare to get a human being born alive with an extra chromosome. There's only a few, like 21, 18, and a couple of others. And we'll go into why that is in a few weeks. But the point here is, here's the chromosome. Notice there are a lot of genes along the chromosome. Notice that we have gene 1, we have gene 2, we have gene 3, gene 4. And what you'll learn this week is that there's, in discussion tomorrow, is that there's sequences at the beginning of the gene that switches the gene on and switches it off. And in the gene, at least the eukaryotic gene, are these magical things, if you've read your article, called introns and exons. And we'll talk all about that tomorrow in discussion and next week. Are there any questions? Yeah, Eden.
Does RNA have polarity as well?
Yes, RNA has a 5' and a 3' end. Always at the end of an RNA is a phosphate at the 5' end conceptually. Absolutely. And we'll talk about that next week. Big strokes, any big conceptual questions? Because we're only, I'm not interested in these little details. What I want you to know is the big stroke. How come a gene has a unique sequence? It has a polarity. Why is the gene unique? How can we manipulate it? What is a gene? I'm not going to really ever ask you about the structure of the nucleotides under the, and I just want you to know conceptually. Okay, we've got to wrap this up. Who's taking charge of this show? Daisy, can you come down here? We have an experiment that I want you to do. And then we'll have another one that you'll look at before you leave. And let me turn the light on for Daisy. And I want you to listen to her. And we have two things going on today. And those two things are the following. Kelly, can you just hold off for a sec, please? Thank you. So the first thing is you all have plates in front of you. And you have little test tubes. And so what you're going to do is a bacterial cloning experiment with your own hands. How many have ever done that? How many have ever handled a bacteria before? Hi? Almost none of you, but some. Faria, have you done it?
Say it again.
No.
Good. So what you're going to do is you're going to do an actual experiment. And it's cloning of bacteria. And there's a number of different kinds of bacteria that you're dealing with, and Daisy's going to take you through exactly what you have to do. So I want you to follow it step by step. Then what I want you to do is I want you to take these little plates home with you. Either put them on the top of a refrigerator or under a pillow so that they get a little bit of heat, because they like to, they're E. coli cells, they like to grow at body temperature. And then what I want you to do is either bring them back on Thursday or keep them over the weekend because some of them may grow slowly, and observe what you get on the plates. And when you leave today, you're going to have a little sheet which is going to have 4 questions, and it's essentially like a lab write-up. It's not very elaborate. It's very simple. And if you hand it in a week from Friday, you have the potential to get 10,000 bonus points, which count in addition to anything else that you get. So Daisy's going to take you through that with the help of Kelly and Aicha, who will be going around the room trying to guide you in terms of what you're doing. And all 3 of these guys are experts at doing this stuff, much more than I would ever be. The next thing is when you leave the room to leave the class, there's a gel here, like you read about for last week's class and for this week's, in which we've taken DNA and we've digested it with a restriction enzyme, or at least we have DNAs of different sizes, and we've stained it with this thing called ethidium bromide. I'll take care of that. Stained it with this thing called ethidium bromide, and you'll be able to visualize using glasses, because it's UV light and protection of your eyes, the DNA as it moved in the gel. So you'll be able to see with your own eyes gel electrophoresis. It's your show, Daisy.
Hey guys, so...
Turn it on.
All right, there we go. Hi, everybody. So you have some things wrapped in tinfoil in front of you. You should all have...
Step by step, as efficiently as we can do it. Okay?
This, you want to unwrap this. In here you have 3 plates with agar. It's really important that you don't mix up the lids and the bottoms because there's going to be a mark on 2 of the plates that tells you what's on the plate. There's 1 plate without a mark, and that's just plain agar and nutrients.
Don't tell them what it is. Part of the lab report.
No, the plates, they know what
Yeah,
it
I know,
is.
but go on.
And there's a plate that has 1 tick mark on it. That has 1 kind of bacteria, 1 kind of antibiotic on it, I'm sorry. And then there's a 3rd plate that has 2 tick marks, and that has 2 antibiotics. So what you're going to do is you're going to label each of the plates. So you're going to draw... Grab a pen. If this is your plate here, you're going to section it into 3 sections. And use the permanent marker that we gave you. I think some of you in the front have to share. And so on every single plate, you're going to section it into 3 and label A, B, and C. And remember to not mix up the lids, please. You should turn it over and do it on the bottom. That's the best way. So you label each plate like this.
Don't open them.
Also on your desk.
Turn them over.
Yeah, turn them
Take
over.
it over. Take them through it again, step by step.
Okay, you have 3 plates. You want to turn each one over and section it into 3 parts like I drew on the board. And you should label a section A, a section B, and a section C on each of the plates. Does anyone have a question about that? The lines, it doesn't matter where the lines are, but they may be on the lid, in which case you don't want to switch them up. Yeah. So once you do that, you all have a rack on the desk in front of you. You're sharing with your neighbor. On this rack, you have 3 cell suspensions. They're labeled A, B, and C. This is the rack that I'm talking about. If you guys all look up really quick. And each of you has 3 tubes. Each tube contains bacteria from a different strain. So they have a different genotype, and they're going to be resistant to different antibiotics, which you should try and figure out based on the growth that you see on the plates. So is everyone done labeling so we can move on? All right, I'll keep going. You guys also have a little tinfoil thing that looks like this. You want to open this up. Inside are sterile toothpicks, so don't get your fingertips all over the ends. What you're going to do is you're going to dip 1 toothpick into each of the 3 tubes that you have. And when you do this, the toothpick is going to pick up bacteria from each of the strains, and you're going to streak it onto each plate corresponding to the section you labeled. So for example, you're going to take out one of the toothpicks, and you're going to dip it into tube A. And then once you do that, where's my pen? You want to streak it into section A. So if this is the same plate here and it's in the same orientation, you want to streak it in this section. And when you're streaking this bacteria, basically what you do is you lightly touch it to the agar, and you just make a motion like that. Don't dig into the agar. Yeah, don't poke the agar. It's unnecessary. You can just lightly scrape it along the surface and it'll...
Remember, flick the bacteria. Flick it so that you mix it up.
Yeah, if you flick the bottom of the tube, if you're holding the tube like this, you kind of can just hit it with your fingers and it'll mix it a little bit. You should do it when the top is closed.
Show me where they're filming.
So once you flick the bacteria, you dip in the toothpick and then you lightly streak it in section A on every single plate.
I don't see it.
And then you can set that aside and do it again with bacteria B and bacteria C. Does anyone have a question about any of that?
You're just replicating the bacteria in each sector of each plate. A bacteria in A sector on 3 plates, B bacteria in B sector on 3 plates, C bacteria in C sector on 3 plates, and you can draw your initials, you can write your name, you can do whatever you want to do, but don't dig in it. You can do whatever you want as long as you don't contaminate it and use a different little toothpick every time. I don't know if there's antibiotics in there. I don't know what the result of this experiment is.
You should only use 1 toothpick per test
I don't know
tube.
why you're going to get the results that you're getting. That's for you to figure out.
So once you guys are done streaking each of the cells, you should have...
How you doing, Dora?
You should have a little piece of
fun?
parafilm.
Yeah.
Did you ever do that in a class before, right in a lecture?
Yeah.
Oh, good. Okay, pretty soon comes the hardest part.
So
you have to demonstrate this, Daisy.
Okay.
Get Aicha to do it. Aicha, put the camera down and let's demonstrate some stuff, please. Okay, you guys ready? When you're done, cover up the bacterial plates. And when you're done, turn them upside down. Always keep the bacterial plates upside down. You don't want any condensation or moisture going on the plate, so you always keep it upside down so that if there's any moisture or condensation, it goes to the top of the lid rather than on the agar where the bacteria are growing.
All right, so Dr. Goldberg says this is the hard part, but this is my favorite part because I like playing with this stuff. You guys have in front of you what is called parafilm.
You just talk and I'll talk. I'll be your voice. Are you going to do it?
No, they're doing it.
Oh, they're going to do it. Okay,
Yeah.
go ahead.
Okay, you have this stuff called parafilm in front of you. There's a paper backing that you remove. That should be self-explanatory. Then, once you do this, you're going to use it to seal the lid and the bottom of your plate so that it doesn't come apart anymore. So if you just all look up, even if you're not done yet, just watch Kelly. You're going to hold it right here and just stretch around all the way.
Don't drop it.
Don't drop it and
Don't
try
break
not
it.
to break
Just
it.
go slowly.
And then you just kind of stick it to itself. So if for some reason you have trouble, raise your hand and we can help you.
How many times have you done that, Aicha? How many? A million?
So once you guys wrap up your 3 plates, you're just gonna, you can wrap it back up in the tin foil and stick it in your backpack or bag or purse and take it home with you to
can
grow.
seal them all together. Show them how to do that.
You show them, Aicha.
We don't have enough parafilm for them to do that. We only have 3 pieces of parafilm.
Ooh, that's not so good. That's bad. Should have brought a whole bunch so they could just put them all together. That's okay. It's okay. How many of you are done? Derek, you done? Okay, those of you that are done can wrap them up. Leave all the other stuff on your little desk. Take those plates, put them in your backpack, make sure they don't open up. Take them home. Leave them upside down, always upside down. If you want, you can wrap them in the tin foil again, if you want. They don't need any light. but keep them upside down. And when you're done, Kelly has another experiment for you to see. Ready, Kelly? Get my stuff out of it. I think they should walk by and see it. If that's okay. Do you mind? Okay. Do you have to take it off there? Or you can. It won't shine through that plastic. You're gonna have to take it off. Won't be as good. Okay. Got it? Did you test it? There we go. Great. Perfect. That you shouldn't have done. That's the way you blow out the UV light. Once you turn it on, you should leave it on. Yeah.
Isn't it bad to like expose it to?
No. It's not going to hurt anyone. Got to leave it on. Let's do that afterwards, Daisy. Once you have, how many are done? Okay. Now, can you turn that lamp on, Kelly? Okay. Can all of you just stand up in your seats? Can you just stand up? Can you see the front? Are you able to see what's in the front? Just from a distance. Are you able, you want to tilt that a little bit, Kelly? Can you do that? You're able to see, you're seeing UV light, and you're seeing some sort of orangish bands. Can you all see that from a distance? You're going to all get a chance to look down close. Those are the DNA. Did you cut it with the enzyme? Yep. Okay, it's digested with the restriction enzyme. You can see, again, like you had in discussion last week, you can see large fragments at the top, smaller fragments going towards the bottom because they have different sizes, and you can see the results of the separation of those sizes using these gels. Now, what I want you to do is one by one, very quickly, come down. Kelly's going to give you a little mask. You just go like this, and you look like that, and you'll be able to see it. And again, you want to just protect your eyes. UV does not go through sunglasses. That's where you wear sunglasses outside, or through this plastic. We have a number of different goggles and glasses and stuff, so a few of you can come up at a time. And then when you're done, feel free to leave with your agar plates and come prepared for discussion tomorrow.